Techniques by separation mechanism. Ion exchange chromatography. Ion exchange chromatography uses an ion exchange mechanism to separate analytes based on their respective charges. It is usually performed in columns but can also be useful in planar mode. Ion exchange chromatography uses a charged stationary phase to separate charged compounds including anions, cations, amino acids, peptides, and proteins. In conventional methods, the stationary phase is an ion exchange resin that carries charged functional groups that interact with oppositely charged groups of the compound to retain. There are two types of ion exchange chromatography: cation exchange and anion exchange. In the cation exchange chromatography, the stationary phase has negative charge and the exchangeable ion is a cation, whereas, in the anion exchange chromatography, the stationary phase has positive charge and the exchangeable ion is an anion. Ion exchange chromatography is commonly used to purify proteins using FPLC. Size exclusion chromatography. Size exclusion chromatography is also known as gel permeation chromatography or gel filtration chromatography and separates molecules according to their size. Smaller molecules are able to enter the pores of the media and, therefore, molecules are trapped and removed from the flow of the mobile phase. The average residence time in the pores depends upon the effective size of the analyte molecules. However, molecules that are larger than the average pore size of the packing are excluded and thus suffer essentially no retention. Such species are the first to be eluded. It is generally a low-resolution chromatography technique and thus it is often reserved for the final, polishing, step of a purification. It is also useful for determining the tertiary structure and quaternary structure of purified proteins, especially since it can be carried out under native solution conditions. Expanded bed adsorption chromatographic separation. An expanded bed chromatographic adsorption column for a biochemical separation process comprises a pressure equalization liquid distributor having a self-cleaning function below a porous blocking sieve plate at the bottom of the expanded bed, an upper part nozzle assembly having a backflush cleaning function at the top of the expanded bed, a better distribution of the feedstock liquor added into the expanded bed ensuring that the fluid passed through the expanded bed layer displays a state of piston flow. The expanded bed chromatographic separation column has advantages of increasing the separation efficiency of the expanded bed. Expanded bed adsorption chromatography is a convenient and effective technique for the capture of proteins directly from unclarified crude sample. In EBA chromatography, the settled bed is first expanded by upward flow of equilibration buffer. The crude feed, which is a mixture of soluble proteins, contaminants, cells, and cell debris, is then passed upward through the expanded bed. Target proteins are captured on the adsorbent, while particulates and contaminants pass through. A change to elution buffer while maintaining upward flow results in desorption of the target protein in expanded bed mode. Alternatively, if the flow is reversed, the adsorbed particles will quickly settle and the proteins can be desorbed by an elution buffer. The mode used for elution depends on the characteristics of the feed. After elution, the adsorbent is cleaned with a predefined cleaning in place solution, with cleaning followed by either column regeneration or storage.